Hello everyone, welcome back to Dr. K. PREM-PRIMER video lecture series presenting by Dr. K. Prem, that's me. As you all know that linkers and adapters are joining molecules. When you want to clone DNA fragments or cDNA fragments with blunt ends, it is not possible. So to generate compatible cohesive ends on DNA fragments, maybe a genomic one or cDNA fragments, then you add these linkers to them or adapters to them. Then you'll get a compatible cohesive ends on those DNA fragments. Then those DNA fragments can be cloned. In the previous lectures, we talked how linkers and adapters being used to generate cohesive ends on blunted DNA fragments. In today's lecture, we'll see the differences between the linkers and adapters. What are the important differences between the linkers and adapters? Both of them are joining molecules, but they have potential differences between the linkers and adapters. Those differences we'll study in today's lecture. You see, linkers are having the internal restriction site and both termini are blunted and have 5' phosphate and 3' OH at each termini like natural DNA. Whereas adapters are having the free-formed cohesive ends. At cohesive end, it is having 5' OH and 3' OH. At blunt end, it is having 3' OH and 5' phosphate. Not only these differences, there are around 10 differences there between the linkers and adapters. We will see in the next table. Here, first column is feature, and the second column is linker, and third column is adapter. So first you see the size of the linker or chemical nature of the linker. You see, they are chemically synthesized oligonucleotide double-stranded DNA. They are double-stranded DNA. And when it comes to the adapters, they are also chemically synthesized oligonucleotide, but they are having a partial double strand, means they have cohesive ends. One end is having cohesive end, other end is a blunt end. And the termini or ends nature, you see, termini or ends nature. Both termini are blunt ends in case of linkers. Both termini are blunt ends in case of linkers. When it comes to the adapters, cohesive end one side and blunt end at other side. Means one termini has cohesive end, other termini has blunt end. Presence of 5' phosphates and 3' OH, both termini have 5' phosphates and 3' OH in linkers. But in case of adapters, cohesive end have 5' OH and 3' OH, and the blunt end have 5' phosphate and 3' OH. So here, in case of adapters, they have two different ends. One is a cohesive end, other is a blunt end. At blunt end, they have both 5' OH and 3' OH, and blunt end have 5' phosphates and 3' OH. In case of linkers, like natural DNA, each termini have 5' phosphate and 3' OH. And the fourth feature is that self-ligation is possible, and in linkers, self-ligation is possible. And there is a chance to form a chain. Chain formation can take place when you are using the linkers as a joining molecules. In case of adapters, there is no self-ligation. There is no possibility of self-ligation of the adapters. And the fifth one is presence of restriction sites. in case of linker, you can have more than one restriction site. In the linker, you can keep more than one restriction site. But in case of adapters, only one restriction site, that too readily ligatable cohesive end. When you're using the linkers, you need to go for restriction digestion after joining of the linkers to the DNA fragments, right? So here you can see restriction endonuclease is required. Once you join this is a linker, this is a DNA fragment. If you join, then it has to undergo restriction digestion to give a cohesive ends. But here, in case of adapters, it is not required because already adapters are have free-formed cohesive ends. Once they ligate to the blunted DNA molecules, and that blunted, then it can be readily used to, it can be used directly with the, we can go for ligation with the vector. And the usage of polynucleotide kinase, in case of when you are using linkers, there is no use of P and K. But when you're using adapters, the cohesive end have 5' OH and 3' OH. So 5' OH is unnatural. Hence, you have to convert that into the 5' phosphate by using polynucleotide kinase. So polynucleotide kinase has to be used after linking the adapters to the blunted DNA molecules, where the P and K phosphorylates the 5' OH of the cohesive end, and that 5' phosphate is being used to adapt, ligate with the vector molecule. Ligation reaction. How many ligation reactions are required when you're using the linkers? Two. First thing is that you have to use linkers have to be attached or ligated with the DNA fragments, and then DNA fragments with the linkers are again ligated with the vector. Two ligation sorry. In case of adapters, also two. Directional cloning is that possible? Means you can control the. Can you control the orientation of DNA fragments being cloned? Yes, in case of linkers, you can. And in case of adapters, when you're using, we cannot do. Success rate means ligations are very, success rate is very high with the linkers when compared with the adapters. Insert internal restriction site effect. For example, blunted DNA have some restriction sites. So such restriction site containing linker cannot be used to attach the DNA fragment. Means you see, linker with such restrictions, such sites cannot be used because linker and the DNA fragment, if they have same restriction site, then when you are going for restriction digestion, it will cut the linker as well as the DNA fragment. So you cannot clone the DNA fragment you want. So that's why internal restriction site and the linker cannot have the same. Linker restriction site must be different when with the insert internal site. Here, adapter with such site can be used. Insert have damage one restriction site. Still, you can use the adapter with the adapter containing the BAMH1 site because there is no, we don't use any restriction digestion to generate the cohesive ends. So adapters have free-formed cohesive ends. So adapter directly attach or ligate to the DNA fragments and then go for the ligation with the vector, right? When it comes to the linkers, when the linker is having, when the insert is having BAMH1 and linker is having BAMH1 site, then when you go for digestion, the BAMH1 digests the both insert and the linker, and that insert, which is become a two parts or two fragments, cannot be used in the ligation with the vector. And substrates, here what we can do is you can use the blunted genomic DNA, or as well as the cDNA. Here adapters also blended genomic DNA or cDNA fragments you can use. So these are the important differences between the linkers and adapters. So this is all about today's lecture. And if at all you like it, try to subscribe my channel. That's Dr. K PREM-PRIMER. Don't forget to subscribe because you miss some updates from my channel when you don't subscribe, right? And if you want to share with your friends, you can. And if you like it, try to give thumbs up so that we'll be knowing who are interested. And if you have any doubt or comment or question, right, or compliment, you can write to me through the comment section. This is all about today's lecture. Thank you all for listening. See you again with one more video. Until then, bye-bye.